In this video, we are going to discuss about domain Archaea or Archaebacteria. So these organisms under this domain Archaea or Archaebacteria form a primitive group of bacteria. They are the most ancient bacteria and are believed to have evolved on primitive Earth billions of years ago when conditions on Earth were hostile. Since then, they have survived unchanged and still live in extreme environments like the Great Salt Lake, Dead Sea, sulfur springs, and volcanic wells. Archaebacteria are known as evolutionary relics or living fossils as they are one of the most primitive group of bacteria found on Earth. So let us look to the general characteristics of Archaea or Archaebacteria. So the general characteristics are as follows. The cell wall is non-cellulosic. The cell wall is non-cellulosic but is formed of polysaccharides or proteins. It is without peptidoglycans. So the cell wall does not contain any cellulose but it contains only polysaccharides or proteins and it is without the peptidoglycans. And the cell membrane lipids are arranged in monolayer. So the cell membrane is of single layer, that is they have a single layer cell membrane or monolayer cell membrane. The lipids have long branched hydrocarbon chains linked to glycerol, which enables these forms to tolerate extreme heat and acidity. So these branched hydrocarbon chains linked with glycerol makes them capable of withstanding extreme condition of heat and acidity. The archaebacterial ribosomal RNA, which is also called the rRNA, has different sequence of nucleotides as compared to true bacteria, that is the organisms which belong to domain Eubacteria. So they are very much different from the organisms and the domain Eubacteria or the true bacteria. Their main difference is of ribosomal RNA, that is the rRNA, which have very much different sequences of nucleotides as compared to the true bacteria. Now next we come to the classification of Archaebacteria. So based on the habitat and metabolic activities of Archaebacteria, they have been divided into three groups. These are the Methanogens or the methane producers, Halophiles, and Thermoacidophiles. And now we are going to take a look on each of the following in details. First we come to the Methanogens or methanogenic Archaebacteria or the methane producer. They are also called the methane producers. So they are obligate anaerobic autotrophs. This means that they cannot survive under aerobic conditions, but they can survive under only anaerobic conditions. So they are called obligate. Now the next point is they produce methane gas. As from the name we can see, they are known as the methane producers. They produce methane gas from CO2 and acetic acid from sewage in marshy condition. So this is one of the important point, the marshy condition. Using the following chemical reaction, CO2 plus 4H2 gives CH4 plus 2H2O and CH3COOH plus CH3COOH gives CH4 plus CO2. So they use either carbon dioxide and hydrogen to produce methane and water, or they decompose acetic acid into CH4 and CO2, that is methane and carbon dioxide. Now the next point is they are found in marshes, bogs, swamps, and in the rumen of cattle like cows and buffaloes. So this last one, the last line, rumen, they are also found in the rumen of cattle like cows and buffaloes. From this point comes the last point, that is they helps in fermentation of cellulose. Rather, they are helping in fermentation of cellulose is directly related to their presence in the rumen of cattle of cows and buffaloes. As we know, these cows and buffaloes eat grass or plants. They are herbivore. They eat only plants. So they need certain specific bacteria in their guts to ferment or to digest the cellulose. So these methanogenic archaebacteria helps in digestion of cellulose in the plants that the herbivores eat, especially cows and buffaloes. Next, we come to the halophiles or halophilic archaebacteria, which are also called the salt lovers. Now, they live in saline environment. As we can see from the name, salt lovers, they live in saline environment like salt lakes, marshes, brine. Brine is basically the solution of salt and salt rich soil where salt content concentration varies from 2.5 molar to 5 molar. The second point is they have high intracellular cell concentration, that is, the cell concentration inside their cell is very much high compared to other kind of cells. Their enzymes and ribosome function efficiently only at high salt concentration. So they live in salty environment or saline environment. As a result, their ribosomes and enzymes function efficiently only at high salt concentration, and their nutrition is chemoautotrophic. Some examples are Halococcus and Halobacteria. Now, the next one is the Thermoacidophiles or Thermoacidophilic Archaebacteria. So they are aerobic or facultative anaerobic chemoautotrophs. This means they can survive both in aerobic and anaerobic condition. Facultative anaerobic means they can survive in anaerobic condition, as well as if the condition changes to aerobic, they are also able to survive in that conditions also. These kind of organisms which are generally anaerobic but can survive in aerobic conditions are called facultative anaerobes or facultative anaerobic organisms. Next, we come to the next point. They are adapted to live in hot and acidic medium, as we can see from the name thermo and acido. So they are adapted to live in hot and acidic medium. Some are adapted to live in very low temperature, even below freezing temperature. So they are not only able to live in hot environment, they are also are capable of living in low temperature environment even below the freezing point. They are found in sulfur springs, example sulfur lobus, in refuse piles of coal mines, thermoplasma, or in geothermal areas, the thermoproteus. So we find sulfur lobus in sulfur springs, thermoplasma in refuse piles of coal mines, and thermoproteus in geothermal areas. So these three are examples of thermoacidophilic archaebacteria or thermoacidophiles. Then they are chemoautotrophs as like the halophiles. Now next we come to the economic importance of archaebacteria. Methanogenic archaebacteria are used in production of gobar gas from cow dung and swage to be used as a cooking gas. So methanogenic archaebacteria are used in the production of gobar gas from cow dung and swage to be used as cooking gas in rural areas, and their heat-resistant thermophilic enzymes and restriction enzymes are used in biotechnology. So the enzymes which are heat-resistant and thermophilic derived from these bacteria, especially the thermoacidophiles, are used in biotechnology. Halophiles are used in bioleaching of pore mines, and Archaebacteria are used as biosensors. So this was all about the Archaebacteria or Archaea. Anyone of you who watched the video till now and have any doubt or any problem, please do comment and let me know about it so that I could be able to solve your problems.